Yeah, I would like to welcome Dr. Aurélie DUTOUR. And hello, Aurélie. Thank you for being here. So Aurélie is a researcher in Léon Bérard Cancer Institute Center in Lyon. And she's after a postdoc in, I think in Houston.
Yeah.
You came back in Lyon.
Yeah.
And you came back again in Lyon because you were in Lyon also before for your PhD.
No, I was in Limoges and Munich. So.
Okay, so actually you are interested in bone sarcoma, and also you are expert in the modeling and the evaluation of new therapy for bone sarcoma. And we are very happy to welcome you, and thank you for presenting your data today.
Well, thank you. Thank you all for giving me thank you to the CLARA for giving me the opportunity to present this work. And actually it was supported in part by funding by the Cancéropôle. And I will share my screen. No, it's not, yeah, here we go. Okay, you're seeing my...
Yeah, it's working. Maybe if you can put it in a plain screen.
Okay.
Okay, thanks.
My main interest is in bone sarcoma, and since a few years ago, we started to get really interested in the environment of these tumors and to analyze it to discover potential prognosis factor and or therapeutic targets. And I'm going to present you part of this work and to get a few background first on chondrosarcoma, because chondrosarcoma is a rare bone tumor. It's the second most frequent bone sarcoma after osteosarcoma. And actually, it's not just one disease. There are many diseases in chondrosarcoma. There is first a conventional chondrosarcoma, which is 85% of the chondrosarcoma, and it's already divided into 3 different grades. And with the increasing grades, the cellularity increases. The presence of chondroid matrix, as you can see, decreases because it's one of the characteristics of this tumor. Each tumor cells will synthesize cartilaginous matrix. So, it's also cell aggregate to form lobulae, and that are separated by septa. And in the end, they create 3 chondrosarcoma. There are no more matrix. And it's a quite invasive disease. As you can see here, we speak about 10 years survival, not 5 years, but it goes down from 80% to 30%. And there are also non-conventional chondrosarcoma, like the mesenchymal chondrosarcoma and the dedifferentiated chondrosarcoma, which is 10% of all chondrosarcoma. And it's quite a weird tumor because it's really composed of 2 different compartments that are just juxtaposed. One is a well-differentiated, often grade 2 chondrosarcoma, and the other one is any kind of sarcoma. It can be osteosarcoma or a soft tissue sarcoma. And these tumors are very aggressive, highly metastatic, and they have a low survival. Why are we interested in chondrosarcoma? Because chondrosarcoma are really therapeutic challenges, because they are highly resistant to chemotherapy and radiotherapy, because they have developed intrinsic resistance mechanism. They express efflux pump. They have a high expression of PGP. Their apoptosis, their resistance to apoptosis. They overexpress BCL2, BCL-XL. It was once even thought to target these proteins to restore kind of chemosensitivity, but without any success. And they present numerous defects in cell cycle. Like, for example, they have deletion of CDKN2A. And their chondrosarcoma have also the extrinsic resistance mechanisms that are more linked to the structure of the tumor, like they have a low vascular density. Vessels can't enter this abundant and dense matrix, chondroid matrix. Vessels are just scattered at the outer edges of the tumor, and they are highly hypoxic. So because of all this mechanism, surgery remains still nowadays the only really effective treatment for this kind of tumor, but the resection has to be monoblock. So it means that the surgeon hasn't to see the tumor. Orthopedic surgeons often say that when they see a bone sarcoma, it's too late, they have gone too far away. And if the resection is not complete, the tumor will relapse with higher grade. And finally, if we start from a grade 2 chondrosarcoma, it will evolve to a grade 3 and then to a metastatic grade 3. So there has been no improvement in chondrosarcoma survival over the last 13 years, despite the improvement in surgical techniques. But nevertheless, we didn't give hope. And with the increasing knowledge of the cellular signaling pathway involved in tumorigenesis, and the development of therapeutic targeted therapy, there was hope that some of these agents could be efficient for chondrosarcoma. And it was thought, and epigenetic targeting was thought in chondrosarcoma, like EZH2 inhibitor, like EDH1 mutant inhibitor, because half of the chondrosarcoma are mutated for EDH1 or EDH2. It was thought to target the cell cycle with PI3K inhibitors, using TKI and pan-TKI inhibitors like regorafenib, and also to target the bone environment by inhibiting osteoclast using zoledronate. But all of these agents were all successful in preclinical trials in animals and in vivo vitro models. But all of this approach, only one was successful when reaching the clinical phase. It was regorafenib with the REGOBONE phase 2 clinical trial, that showed a non-progression in 54% of the chondrosarcoma patients enrolled in this trial and getting regorafenib. So what does all of these approaches tell us? That maybe it is time to think outside of the tumor cell and consider chondrosarcoma immune environment. Well, because it is well known now that a tumor in itself doesn't, well, it interacts with its environment for most of it with the immune environment. And this immune environment is composed of immune effectors that will kill directly the tumor cells, like the effector T cells, the NK cells, some that will have an indirect activity by helping the TH cells, the M1 macrophages, and that will, all this effector will either kill the tumor cells directly, or they will also create an immune stimulative environment. But it's not so simple. There are also immune suppressive actors in this tumor environment, like the Tregs, that will halt the activity of the effector T cell, the M2 macrophages, and all these immunosuppressive will help create a pro-tumoral environment. And the goal of all the immune therapy is to go from this pro-tumoral immune environment and revert to the anti-tumoral immune environment by stimulating the effector cells or turning the macrophages from M2 to M1 by getting a shift in this. The problem with the chondrosarcoma is that chondrosarcoma was long thought to be an immune desert. And we started a study a few years ago where we mapped the environment, the immune population present in a cohort of conventional chondrosarcoma. And we showed that there was some immune cell located at the periphery of the tumor mainly, and that these cells are linked to tumor progression, that there are more macrophages, CD163 macrophages than T cells, and that the ratio CD8, CD163 is indicative of the aggressivity of the tumor. The higher this ratio is, the higher the survival of the patient is, and the less aggressive are the tumors. And in a rat immunocompetent chondrosarcoma model, we were able to show by doing a selective depletion that CD163 macrophages have a pro-tumoral activity, that they help the tumor to progress, while CD8 T cells are anti-tumoral. And there was what was known till a few years ago, like 3 or 4 years, regarding the ICP. It's the only one that was studied was was PD-1, PD-L1, and PD-L1 was found expressed only in the differentiated chondrosarcoma. It's absolutely not expressed by conventional chondrosarcoma, and it has no prognosis value. So with this, we went on and gathered a cohort of conventional and also the differentiated chondrosarcoma, where we decided to map the immune population by EHC. We decided to use technologies that can be used, techniques that can be used in everyday diagnosis by almost every lab. So we use CD3 to map the TILs, CD8, CD68, CD1, and 63. And we studied the expression of immune checkpoint by qPCR, activator or inhibitor ICP. And the one that was most relevant, with which expression was the most relevant, was validated by EHC. So in this conventional and dedifferentiated chondrosarcoma, we saw that there are immune cells that are CD8, CD68, and CD163. That in conventional chondrosarcoma, these cells are located at the periphery of the tumor, but they are also they can also sometimes infiltrate this the septa that we can find between the lobula formed by the tumor cells. And in the dedifferentiated chondrosarcoma, these cells are located also at the periphery of the tumor, but they are really infiltrating the dedifferentiated part of these tumors. And macrophages are the most abundant cells present in this environment. The composition of this immune environment is not correlated with patient age, gender, or the tumor location, but it's correlated with patient survival. For most of it, for the presence of CD3 T cells, patients with a infiltrate low in CD3 T cells have a poorer survival than patients having a higher density of CD3 T cells. And looking at the total macrophages, the same was observed. A high density of macrophages is associated with a poor survival. And the most interesting thing is the ratio between total macrophages and effector T cells. The patient having a low ratio have a really better survival. That's in multivariate analysis is really significant, while the patient with a high ratio of CD68 over CD8 have poor survival. So in all chondrosarcoma, like in most of the bone sarcoma, macrophages are the key players in this immune environment. And the balance between macrophages and T cells is indicator is a prognostic value, was indicator of tumor aggressivity. As I said, tumor effector cells are not the only component of the immune environment. They are also the immune expression of the immune checkpoint. And this was analyzed in conventional chondrosarcoma and differentiated chondrosarcoma, for which we had material. And by looking at the expression, we saw a high variability and between grade 1, grade 2, grade 3, and differentiated chondrosarcoma. Here, the checkpoint, the immune checkpoint that seemed of most interest for us were B7H3, CSF1, CSF1 receptor, PD1, PDL1, and SIRP alpha. So we looked at we try in a matter to try to score the expression of this immune checkpoint in the cohort. We compared it with the expression of this same marker in cell lines known from databases to be negative. Here, for example, for the B7H3 is a Kazumi cell line, and one known to be positive. And we saw that there are also that PD-L1 is absolutely not expressed in conventional chondrosarcoma, while it's expressed in the differentiated chondrosarcoma. That CSF1 receptor is expressed similarly in both tumor chondrosarcoma subtypes, and that TNF-alpha is expressed mainly in conventional chondrosarcoma. When we think of the famous PD-1, PD-L1, we looked at the expression by EHC, and we confirmed that it's absolutely not expressed in conventional chondrosarcoma here, and that it's present, really expressed in the differentiated chondrosarcoma, just in the differentiated part of these tumors, and that it has no prognosis value in the differentiated chondrosarcoma. Well, the two checkpoints that were common to conventional and differentiated chondrosarcoma are BCVNH3 and CSF1 receptor. So we focused on CSF1 receptor because of its implication of in macrophages and because of the importance of macrophages in chondrosarcoma. And we showed that we confirmed by EHC the expression of this immunomodulatory molecule by macrophages in both chondrosarcoma subtypes. Its expression increases with the grade, but it's expressed in more than 80% of the differentiated chondrosarcoma and more than 50% of the conventional chondrosarcoma. So what this study shows is that macrophages, the chondrosarcoma are really not the immunodesert that's what it was thought to be a few years back, that macrophages could be a therapeutic target because they are the most abundant population, and they are linked to tumor aggressivity, or they are a prognosis factor. So we have to find a way to shift this balance toward the presence of more CD8 T cell, or to make the macrophages move from the M2 to the M1 phenotype. And as I said before, in the rat model, we showed that mifamurtide, that's an immunomodulator agent moving macrophage from M2 to M1 phenotype, can slow tumor progression. And now we established that CSF1R could be a potential target for chondrosarcoma, and that's something we are now investigating. I thank you for your attention, and if you have any questions.
Thank you very much, Aurélie, for this nice talk. So again, maybe I can ask people in the audience, do you have any question? Maybe Dr. Zhang?
Hi, Hi, can you hear me?
Yeah, because I was thinking maybe you want to address a question?
Yeah, sure. I will also be getting ready to speak, but I do have a question. This is a very interesting presentation, but in my next talk, I also happen to cover some overlapping areas. So I just want to ask about the CD163 positive macrophage cells. When you're studying such a group, did you try to do this more detailed subtyping of the cells to see if all the cells are equally sensitive to anti-CSF1R?
Well, in the study I showed you, we didn't try any CSF1R inhibitor. That's something we're investigating now. And we didn't look at the density of intensity of expression, as we just did it by EHC. But it's really interesting yet to look at. I could go back to the data from the qPCR and look at the variability of CSF1 expression between the patients. Yeah.
All right, great. I hope you had time to stick around for my talk, because I will also be talking about this a little bit.
Great.
And actually, Aurélie, I have a question. Actually, you say that this sarcoma is very well identified as very rich in extracellular matrix, and you showed that macrophage is also very important and might be something important for this tumor. And do you know if macrophage can find something very important in the extracellular matrix to have the opportunity to be attracted, or I don't know, something?
Well, that's something we, you know, we in bone sarcoma immune field, we come from a long way back. We are really not advanced in comparison to all the other tumors, but that's something I'm thinking about to look at, you know, the interaction, the expression of the matrix and the presence of these macrophages, yeah. Because that must be something.
Yeah, and can you tell us a little bit more about CSF1R? What do we know about this molecule?
I haven't, I must say, I have shifted from this project to another one since the last 8 months. So I'm not sure. Right now, I won't be able to tell you tons about CSF1 receptor. Sorry.
Okay. So thank you very much anyway. So there is no, I'm looking at the chat, no question. And actually, so now it's the turn of Dr.
There is one question going in the chat.
I didn't see.
Isham Mertani. In human clinical samples, do you have information about the mechanisms that regulate T cell and macrophages access to the bone tumor?
No.
Is it possible to increase it by any means?
We don't have this information because all the clinical samples we have are FFPE, and that's something we should. How can we look at that in just the FFPE? But Isham, we can talk about it. I would be happy to talk about it, and that's something yeah, we have to look at. Find a way to look at, and in the samples I have, no, because some of them have also been decalcified. So, you know, doing many experiments or many investigations on these samples is not so easy.
Thank you.
Okay, thank you again, Aurélie. Thank you very much. Thank you for your participation.